This lecture will continue our discussion on methods of phylogenetic inference. We're also going to talk a little bit about alignment of characters. And then we'll but show you an example of matrix and the problem on the worksheet and how you can work on that and how you'll do that problem on the worksheet. So let's jump in. We're going to start with alignment. Alignment is basically a statement of homology at the genetic level, but you could also do it with morphological characters. And on our worksheet, I'm not going to make you sequence DNA. It's a little bit technical, the sequencing, the alignment of DNA, but with characters we can do it rather easily. But the gist of it is that along the left-hand line of our matrix, we have species. So here they don't have their proper name, they're just A through H. And then each column in our matrix represents a position in the DNA or a specific character. So if this was the color, for instance, we'd say A represents, they all have the exact same color. But in this case, it's DNA. At that position in the DNA, they all have an adenine. And so we try to make an alignment so that in each column, we can compare, so to speak, apples to apples. So we're not saying, hey, let's look at color here and then, blood type, and put color and blood type together. You don't want to do that. You want to make each of your characters separate. And so a DNA alignment allows us to investigate every single position in the DNA as a potential character. And so once we were able to sequence DNA, it was readily apparent that this would be a very rich and valuable resource for phylogenetic inference. So for instance, character number 1, which they have highlighted here, they didn't mark the first 2 columns because they're all identical. There's no information there. But notice that species C and D are different at this position of their DNA than all of the other species. And so that is a potential bit of evidence. It's a potential synapomorphy, but we won't know for sure till we map all of our characters onto it, find the very best phylogeny, and then we'll be able to tell if it's a synapomorphy. Now, on this phylogeny, notice it is a synapomorphy. C and D are most closely related. Boom, it's right there. And in fact, they've mapped all of these characters on there as nice, single, simple synapomorphies, but there's always in real data going to be some characters that disagree, some that are convergent or some that are simplesiomorphic. Now, doing a DNA alignment can be very, very tricky. If there's lots and lots of diversity and variety, it can be difficult to find those statements, and then each of your columns may not represent a base pair because we can have mutations, and we're going to talk about different types of mutations in Unit 2. Okay, but mutations make it very tricky to do an alignment. Also, if your DNA gets longer or shorter, some mutations you insert DNA or take away DNA characters, those base pairs, that makes alignment difficult. Now, as genetic distances get more and more and more different, our alignments become less and less tractable, less easy to do. And at some point, we basically lose any semblance or any idea of being able to compare. They're so diverse and so different. So in some ways, it's kind of like saying, if you were untrained in morphology and I said, hey, here's an earthworm and here is a beaver or a rabbit or, any other complex mammal. And I said, hey, compare the earthworm and the rabbit. You would look at it, and as an untrained morphologist, you'd be like, they have cells, they have muscles, but it would be really, really hard for you to make many comparisons because they have changed so much. Now, at one point, the rabbit and the earthworm shared a common ancestor, and they actually both belong to the kingdom Animalia. They have muscles, but if you're going to say which muscles line up, so which muscles in the earthworm are descended from the same muscles in an ancestor as the muscles in a beaver, you might have a very difficult time doing that because it's changed so much. And the same thing happens at the DNA level. If DNA changes so much, so rapidly, it can be very difficult to establish this alignment and these homologies. Okay, so in this class, we don't have to worry about that. As long as you know what alignment is and that it can be very difficult as DNA diverges, that's the level of understanding you need to have. It's not, we won't get into the details. If you're really interested, you're weird, but no, if you're interested in this, I teach an upper division course in phylogenetic systematics, and we talk a little bit more about alignment, and we even look at some of the programs that have algorithms that allow us to do these DNA alignments, okay? But what ends up happening is as we count differences, we realize that some species are more similar to others. Like A and B may be very similar and they may only have a couple differences. But if I compared A to H, then I might see way, way, way more differences. And so we call that a genetic distance. A genetic distance is simply counting up all the differences between an organism and figuring out how many there are overall. And we can measure these at the DNA level, which is the primary way that we do it. But you could also look at, does it make a difference at the protein level? Because that's not always the case. Sometimes a change at the DNA level means that the protein is still the same. So TAG codes for tyrosine. If we mutate TAG and make it a TAA, thymine, adenine, adenine, then it's actually still the same amino acid. So even though we changed the DNA, we didn't change the protein. Okay. Now, at this point, we are going to talk about some other methods for phylogenetic inference besides parsimony. Parsimony we have defined, and we're actually going to do an exercise with it at the end of this discussion. It's on the worksheet, same one on the worksheet, to help you kind of look and get an example of how parsimony analyses are done. But the other ones, we're not going to look at the mechanisms of how to do them. As long as you know there are other methods and they have their strengths and weaknesses, we'll leave the details of that to another class if you want to go on and take an upper division course in this, okay? But one of the methods, which I don't have listed here, let's go ahead and put it on it, because it uses genetic distances. And this is called neighbor joining. Now, in neighbor joining, I can't spell, we take the genetic distances and we take the species that are most closely related and we put them together. And then we add on the next closely related species and so on and so forth. It's a bit more technical than that, but we basically add them on. Now, neighbor joining has strengths and weaknesses. There's a short paragraph in paper number 6. You should take a look at that. That'll give you a little bit more information about neighbor joining, but we don't need to know much more than that. Okay. There are other methods that are also use distance. So there are other distance methods that are a little bit more rigorous and allow for going back and revising any errors. So if you put two species together in a neighbor joining analysis, they're there and they can't be separated. And other ones we can go back and reevaluate and do them a kind of in a more iterative process. But as long as you know that neighbor joining is a distance method and there are other distance methods, that's all you need to know. And then the last class of methods that we're going to talk about, but not in a lot of detail, are statistical methods. And statistical methods arose because of a question we posed earlier in our last discussion. So remember, we asked you if all characters are treated equally. And parsimony does. It treats every character you put into your analysis the same. Everybody gets the same vote, so to speak. So if character 1 says that, species A and B should go together, and character 2 says no, species A and C should go together, you need more data to kind of break the tie because they're both evenly weighted. However, people realized, especially as they gathered these large DNA data sets, that there was additional information in those data sets that we could potentially use to help us determine if we really should treat all characters equally. And so basically, this isn't a good thing, but imagine that there's some negative U.S. history in this, right? But imagine that some people got more of a vote than others. It's kind of the old joke that, which I guess it's a joke, it's kind of sad but true, is that everybody's equal. Some people are just more equal than others. I think that's actually a quote from Animal Farm, where all animals are equal, but some animals are more equal than others. But imagine, we won't get too deep into the politics here. Imagine that some characters, right, have more weight or they're a little bit more trustworthy than other characters. And if you had evidence to back that up, then you could use that in a statistical approach in your analysis. So for example, if you notice that your DNA has many, many more adenines than thymines, then you could use that to help inform if a change to an adenine or a mutation to an adenine is a different value, a different weight than a mutation to a thymine. So maximum likelihood and Bayesian analyses use statistical approaches with these measurements that they take from the data to inform the analysis. So some changes are better than others. So for instance, if you have two characters, one that says A and B should go together, and another one that says A and C should go together, if you trust the character that said A and B should go together more than A and B, then you now have a way to make a decision without adding additional data. And so that can be useful and can be helpful and informative. And most in-depth analyses today rely on these statistical methods. You don't even need to memorize the names. Just know that there are other statistical methods. Maximum likelihood and Bayesian are the two most common, the heavily used ones. And they are using these large matrices, right, gathered from DNA sequencing data and alignment. Okay, and we end up with these nice, beautiful phylogenies, giving us well-supported hypotheses about relationships of organisms. Now, whatever method that we're using, we need to evaluate multiple hypotheses. Now, the best way, the most complete way to do this would be to evaluate every single possible phylogeny. So if I was to do a parsimony analysis, all I would need to do is say, well, let's look at every single possible phylogeny for these species. Maybe we have 6 species in our analysis. And just map my characters, as we'll look at in the worksheet and as we kind of have explained in this lecture. Map them onto that phylogeny, and then whichever one is the shortest, right, has the least length, right, I have to map the least number of characters onto it. That's the best one. That's a very easy, straightforward criterion for picking our best phylogeny, right? However, there gets to be an issue. We cannot, even with the fastest supercomputers, analyze every single possible phylogeny unless it's for a really, really small analysis with just a few species. And here's why. This is for unrooted trees. The number's actually a little bit bigger for rooted trees. If we have 5 species, we have to evaluate 15 different phylogenies. There are 15 different ways to arrange 5 species in a phylogeny. And you're like, okay, fine, that's easy. We can do 15. And in fact, this is all implemented on computers nowadays. So 15 you can do in just, milliseconds. But this number grows exponentially, and not only does it grow exponentially, but it grows exponentially very, very fast. And so by the time we get to 30 species, we would need to evaluate. So there's millions, billions, trillions, zillions. I don't know what comes after, right? We're not quite up to a Google, which is a one with 100 zeros after it. But we get there actually fairly quickly. So with 50 species, we end up with something like 10 to the 85th power of different phylogenies that we would need to evaluate. And our brains are not very good at, realistically estimating and understanding numbers that big. But just to kind of give you a little bit of impact here, just for a frame of reference. How many molecules are in the visible universe? I know, this is why we have Google, right? People have estimated this. So somewhere between the atoms, right? It's a better way to say it than molecules. But 10 to the 78th or 10 to the 82nd. So by the time we get to 50 species, there are more phylogenies to evaluate than there are atoms in the visible universe, which is just this massive, incomprehensible number, right? So the short take home is that we cannot evaluate every single possible tree. So how can we be certain that we found the very best one if we can't evaluate them all? Well, basically what we do is we do a algorithm that allows us to do a different search strategy. And I'm going to give you an analogy. We're not going to get into the details of how those algorithms work, but we don't search every single possible tree once we get up to 30, actually probably even fewer than that. But once we get a reasonable number of species in our analysis, we don't evaluate every single possible tree. But we use an algorithm. And here's an analogy that might help you kind of understand the approach that people are taking when they design these algorithms. Imagine I asked you to find the highest mountain in North America, okay? But you had a limited array of tools that you could use. So you have a whole bunch of maybe you have like 10 people and they all have altimeters. And you're on an unknown continent, you're asked to find the highest mountain. Now, you get to North America, you arrive, I don't know, maybe you make landfall in Florida. You look around, how much time are you going to be spending looking for the highest mountain in North America in Florida, right? Are you going to measure the place where you land and then take one step and do another measurement, and one step and another measurement? You kind of do this circular grid-like pattern and you measure every square inch of Florida and you're like, okay, My highest place is, I don't know, like 300 feet above sea level. I don't know how high the highest place in Florida is. Yeah, that's a waste of time, right? So there are other strategies where you can be reasonably certain you found the highest peak without having to measure every single point in North America. So anyway, there are similar strategies that we can use as we're measuring the number of trees or phylogenies that we need to evaluate to find the very best one overall. But it is still a very long and involved process. And so often people will use big, huge supercomputers with lots of power to help them find the best phylogeny. But again, they're not searching every single possible one, but they can still be reasonably certain that they found the best one. Okay, so we implement a search strategy that does not, it's not an exhaustive search strategy, but it still is good enough. Okay. And then after we find our very best phylogeny, whether we use parsimony or neighbor joining or one of those statistical methods, there are other things that we can do to help us determine whether we have more support and we're more confident in some parts of the tree than we are in others. And in fact, if you look back to this phylogeny, there are numbers above and below some of the different nodes. And a value of 100 means we're very confident in those relationships. If it's below, like especially as it gets down close to 50, that means we're not really so sure about that set of relationships, that maybe it's, not great. And sometimes if it's below 50, they'll even collapse the node. So here we have a polytomy where we have 1, 2, 3 branches that all are coming from that common ancestral node. Okay, so if you see numbers above a phylogeny, just know that it's a support measure. Bootstrap support is one of those. Again, we're not going to get into the details of that because it's beyond what we need to do for this class. But there are ways to do additional analyses to determine whether we're more confident in some parts of the tree than we are in others. Okay, so now as you are looking at a phylogeny like this, you should be able to start to feel more comfortable, maybe even identify. If I gave you a matrix, we won't do it for one this big, but we do have a smaller version on the worksheet we'll look at here in just a second. But if you were given a matrix, you could map characters onto this to determine whether this phylogeny is better than maybe a competing hypothesis. So if we're going to map the origin of feathers on this phylogeny, theropod dinosaurs all had feathers, and our birds are the only living remnant of that clade. So we map feathers on here one time. I can put a branch. Let's go ahead and do it. Let's insert another shape, same one, and I'm going to put it right there, right? I'm going to map feathers onto that phylogeny in one location. So feathers evolved one time. Extinct organisms like this, Velociraptor had them. Birds still have them, right? Now, is it possible that feathers evolved twice? So for instance, go back here. What if instead of mapping this on there one time, what if I mapped it on there twice? So I'm put it once here, and then, oops, let's just copy and paste, that'll be easier. And then once along that lineage. Is it possible that feathers evolved twice? Well, it is, right? But it's less likely. It's not as elegant a solution as having it evolve once in an ancestor. And so this is the one we prefer under parsimony explanation, right? And unless we had some very dramatic other evidence outweighing this one, we would infer a single origin even under those statistical methods, okay? Anyway, so this is just here. Again, we'll probably come back to this. You might see this exact same phylogeny on a test. You should be able to answer questions about synapomorphies, about monophyly, about homology, convergence. All of those things are going to be done, and we're going to get some more time and practice and exercise with those things. So let's take a look at worksheet number 1. On worksheet number 1, there's a problem. It's problem numbers 3, 4, 5, and 6, and 7. Some of it's just vocabulary and making sure you know the definitions. I'm going to zoom in a little bit here so you can see it. Okay, so you are asked to build a matrix, and then using the matrix you've been asked to determine whether this set of relationships is better than this one. Now, in reality, there are many more relationships than this. This is 5 different species. We'd have at least 15 unrooted. It's something like 25 rooted trees, maybe fewer than that. But anyway, a lot of trees that you would have to evaluate if you wanted to do every single one, but we're only determining is this one better than this one or vice versa. Okay, so we're not doing an in-depth analysis. Now, I don't care what characters you choose. If you are a scientist, you might have some good ideas, and you might even choose to not use some characters because they're obviously not very good. So if I wanted to say, is it cute? That, if I was doing analysis, that's a really bad character to use because it's a subjective decision, right? We can't objectively say yes or no, and it's not a great one. But if you want to do that for this analysis, that's fine. I don't care. But maybe think about, if approaching this as a scientist, look at your organisms and see if you can think of characteristics that might be useful. So another one, I'm just going to show you an example. It wouldn't be good. Does it have a backbone? And you're like, wait, that's kind of scientific. Why isn't that a good character to use? Well, look at this. Chickens, platypus, kangaroo, sea lions, cats, they all have backbones. So it doesn't help us decide relationships because it's a much more distant ancestral one. If we had earthworms and scorpions on here, then maybe backbone's a good characteristic. But in this data set, it doesn't help because it doesn't help us separate organisms. So ideally, we're looking for characteristics that are different, right? Some organisms have different states than others, and help us to separate more than one species away from the others. Because if I just say, so for instance, let's say I'm going to say, does it have poison spines? You know this, by the way? Male platypus have poison spines. Yeah, is that kind of cool? Anyway, they're the only, I guess I should say venomous, because I don't know if maybe if you ate them, it would be a problem too. But they're venomous. They're meant to be a defense against predators. So they have venomous spines. So we could be more correct here and say venomous. Now, that's a cool characteristic, but is it useful in helping us decide what the platypus is related to? Well, all it does is say, well, platypus is different than every other species. We would eventually map it somewhere along that branch, but it doesn't tell us what they're related to. Now, again, for this exercise, that's actually fine. I don't care what characters you choose. You can choose anything you want for these 6 characters, and you will then need to make a matrix out of them. So let's do one that I'm going to be a little bit more serious about. Let's say, does it lay eggs? Okay, that's going to be our character. Now, character number 1, I'm going to go for each of my species, and I'm going to put, you can use plus minuses, you can use checks and X's, but typically in analyses like these, they use a 0, 1, just like a binary character state. So if it lays eggs, I'm going to put a 1. If it doesn't lay eggs, I'm going to put a 0. A platypus lays eggs. A kangaroo does not lay eggs. Cats and sea lions do not lay eggs. And when I say lay eggs, I mean their egg is external and development occurs externally. Because kangaroos, cats, and sea lions, right, all have eggs, but they're internal. They're fertilized internally, and development is internal. So I now have a character that helps me decide which of these relationships is better. So if I want to put this character onto this phylogeny, notice that on this phylogeny, I can put that in one place. So let's make a map. And Word is not ideal for this, but it works, and I'll show you how to do it. If you'd rather do this by hand and just draw it out and then take a picture and upload like a JPEG or PDF for the worksheet, that's fine too, but you can do it in Word. So let me show you. I'm going to insert the same thing, a shape. We'll use this nice little square. Notice that if I draw a line across that branch for character number 1, and maybe even I will copy and paste it just to help me remind, I'll color code them. So I'll put this little blue bar up there to remind me that character number 1 is a blue bar. Okay. Notice that if I put it across that branch, I have separated on one side all of the things that lay eggs from all of the things that don't lay eggs. So the simplest explanation on this phylogeny for why the chicken and the platypus can lay eggs is they lost the ability to lay eggs one time. Now, is it possible that egg laying evolved separately in the chicken and the platypus? So I mark it there and there. Well, yeah, that's possible, but that's not the best explanation on a parsimony criteria, so we put them in one place. Marking them twice is kind of like inferring the FBI framed your dog, right? Or maybe, in theory, you could say, well, eggs evolved in the ancestor, and then were lost in the chicken, and then were regained in the chicken, and that's why the chicken can lay eggs, because they were we gained the ability to lay eggs, we lost it, and we gained it back. We're getting into kind of conspiracy theory territory, right? So we prefer the simplest explanation. Sometimes the simplest explanation is, well, we have to map it on there twice. So let's look at eggs here. So there are two explanations for, actually there's only one simple explanation for this phylogeny of why the chicken and the platypus can lay eggs if this is the correct set of relationships. So let's map it on this phylogeny. The chicken and the platypus gained the ability to lay eggs separately. So now I've drawn lines again, separating all the egg layers on one side of the lines from all the non-egg layers on the other side of the line. And so I've defined that character, but this is a more complex explanation than if this is the set of relationships. So if that's my only character, I've got to put my blue bar back up here so I remember which is which as I move on. If that's my only character, this is a better explanation than this one, but we're going to map all of the characters. So let's move on to character number 2. So let's say character number 2 is, do they have well-developed canines? And again, you can use whatever characters you think are useful. Okay, so sea lions, you ever seen a sea lion? Again, you might not, you might have to Google this. That's why we have Google, right? Sea lion teeth. See if we can get a nice picture here. So there you go. You can see them right there. See those canines? Those are pretty well developed. Cats have nice big canines also, so they're their canines. There's a nice one view right there. Boom. So yeah, well developed canines. So we would map that character on here for the sea lion and the cat. Kangaroos do not. Platypus don't. Platypus don't have, I don't know, maybe they have small teeth in their bill. Chickens don't have teeth, obviously. So we would map that well-developed canines characteristic. We'll pick a different color just so we can distinguish it. So maybe we would do this. For character number 2, I'm going to use, shape format, fill. Let's just use a nice red fill. Okay, so red with a blue border. That's pretty. So I can put it on the phylogeny here. And here. Now notice both, there's a simple explanation. So this is not really a useful characteristic in helping us determine between these two trees, but that's fine. Some of them are going to be like that. And so well-developed canines, the chicken doesn't have it. The platypus doesn't, the kangaroo doesn't, but the cat and the lion do. Okay? So 2 characters now. This is still my very best overall hypothesis. So your challenge now, you have 4 more characters. Pick whatever you want. Right? Maybe try and think of things, but if you want to do color, if you want to do, distribution, where do they live? Do they live in Australia? Yes or no? Anyway, whatever you want to do, use. Try and come up with characters. I really don't care too much what the characters are, as long as you map them correctly and then use that to determine which is the best tree. So as this stands, this is the best tree. Now, by the time you get done with all your characters, you might very well have the same number of characters mapped on tree number 1 and the same ones on tree number 2. That's actually okay. If you did that in real life, what you would want to do is you would want to gather more data. But for this exercise, that's fine. So if you had the same number of characters mapped on both trees, then you would say, because this question is asking you, using a parsimonic criterion, which phylogeny is best? In this case, I would say the one on the left, because it's a simpler explanation. But if I had 8 characters mapped on this tree and 8 of them mapped on this one, and they're even numbers, then I would say they're both the same because both of them are equally complex or equally simple. So there is no correct answer. Well, there are correct answers for this, but different people are going to have different correct answers because they will build their own matrices that are going to be different. Okay, and again, I'm happy to go over them with you if you want. You can meet me on Monday, Wednesday, Friday in that Zoom meeting, or set up a meeting to go over your worksheet with me if you would like to. Now, the last few, let's go over these rather quickly. List a physical feature that is a synapomorphy for the group Mammalia. Remember, synapomorphy is an ancestral characteristic that they all share. No one has lost it. So we could put hair. Now, you have to be careful because whales, they do have hair. Maybe a better term would be follicles. They still have those follicles. They're a little different, modified, but all mammals have that. But easiest ones are often, again, the main big groups are named after synapomorphy. So mammary glands is a nice, easy go-to one for a group Mammalia. That's a synapomorphy. All mammals still have mammary glands, the females of the species. So mammary glands in females, that would be a characteristic. That's a nice synapomorphy for mammals. There are many, many others. Mammals is a well-defined group with lots of synapomorphies, but you just need to know one. Okay. Now let's list a physical feature that is a symplesiomorphy for the group mammals. Okay, so something that the ancestor of mammals gained, but has been lost in some of the descendants. Now, this can be a little bit tricky because there are some symplesiomorphies that are really good for the group, but also apply to other groups, right? So if I said walking on four legs, that's a symplesiomorphy. The ancestor of all mammals walked on four legs, but some mammals have lost that quadruped form, right? They no longer use all four legs to walk. So that's actually a good feature, but realize that's occurred in other organisms also. So in the dinosaurs, right, their ancestor walked on four legs, but some of them lost that. So that's a good synapomorphy for multiple groups, or sorry, symplesiomorphy for multiple groups. But that's okay. I'm going to be a little bit flexible on this one. But you wouldn't say backbone, right? That's not a symplesiomorphy because all mammals still have backbones. So even though backbones is much more ancient than just the mammals, it's not a symplesiomorphy for mammals. They all still have it. Now, a really good one, if you want to use this one, that's fine. But maybe try and think of some others, something that all mammals had at one point. So their ancestor had it, but some of them have lost. Complex teeth is actually a pretty good one. The early mammals evolved a diverse types of teeth. So they have incisors and canines and molars and eye teeth, all these things in between, right? Most mammals still have that at various levels. Some have, more complex teeth than others. However, some mammals, including whales and dolphins, lost this complex teeth, and they actually have teeth that are a little bit more like the ancestors. Ancestral teeth were very similar. One tooth was pretty much the same as the one behind it and the one in front of it. And if we look at dolphins and whales, they have that characteristic again. So they have these simple peg-like teeth, right? So that's maybe a good symplesiomorphy because the ancestor evolved it, but then it was lost in some of the descendants. Okay, last one. Symplesiomorphy, and this one should be easy, hopefully. Think about it. What is homoplasy is where you have a more complex explanation. Either was gained and then lost in some descendants, that's symplesiomorphy, or it was gained independently in two different groups, right? And we call that convergence. Okay, so that should help you. Good pointers on numbers 4, 5, and 6 in the worksheet.